Hi, I'm Bhaskar, and today I'll be going through the Graduate Aptitude Test for Biotechnology 2022 question paper along with you. So, this paper has two sections, section A and B, with the B part particularly focusing on biotechnology. And section A contains general aptitude questions, including questions from physics, chemistry, mathematics, and biology. So, I'll be starting with the biology section of biology part of section A first, and this starts from question number 46. So the question number 46 states that given below are two statements. One is labeled as assertion A and the other is labeled as reason R. The assertion is hematopoietic stem cells are considered multipotent precursors, and the reasoning is that they have the ability to differentiate into different type of blood cells. So as the assertion says, it is a true statement that hematopoietic stem cells is a specific type of stem cells which gives rise to various precursors of the blood, different types of precursors in our blood cells. These form the general granulocyte precursors and monocyte precursors and lymphocyte precursors. Hence, it is known as a multipotent precursor. Therefore, the state assertion A is correct, and the reason for for it to be known as a multipotent precursor is that it gives rise to different types of blood cells, and therefore the reasoning for the assertion is also correct. Hence, the option 1, which says that both A and R are correct, and R is the correct explanation of A, is the correct option here. Moving on to glycosidic linkages. So, the question number 2047 asks that what are glycosidic linkages and what are the what kind of bond it forms between two molecules. So, the options we have are sugars, nucleotides, amino acids, and lipids. First of all, a bond joining a carbohydrate with another carbohydrate is known as a glycosidic linkage. So this is a correct option. Coming to the rest of the three options, nucleotides are joined together are by hydrogen bonds and dinucleotide linkages, whereas amino acids are joined by peptide bonds and lipids are joined by ester bonds. Therefore, the correct option for us in this case is the first option, sugars. Question number 48. What is the bond that can form between two cysteines in a protein? A. Ionic bond. B. Hydrogen bond. 3. Disulfide bond. And 4. Amide bond. So these are different types of bonds uh, present in protein interactions. A specific bond which forms between two sulfide atoms of different amino acids is known as a disulfide bond. This can only occur in between amino acids which contain sulfur as one of its constituent atoms, and there are only two amino acids which are known to have this. One is cysteine and one is methionine. Therefore, between two cysteines, the most common bond to be formed is the disulfide bond, which is our option 3 here. 49. Cystic fibrosis is caused due to 1. Mutation in gene encoding G protein, 2. Deficiency of adenyl cyclase, 3. Mutation in gene encoding an ABC transporter, and 4. Overactivation of calcium ion ATPase pump. So, cystic fibrosis is caused due to mutation in the CFTR gene. First of all, we should know what is the CFTR gene. It is CFTR protein is part of the ABC class of transporter proteins, and a mutation in CFTR gene causes cystic fibrosis. So, like you can see, the third option which mentions that the mutation in gene encoding ABC transporter, this is the correct option. Question number 50. The primary enzyme responsible for the rapid and accurate replication of genome in E. coli is. So, in bacterial replication, there are 5 DNA polymerase enzymes involved. DNA polymerase 1, 2, 3, 4, and 5. Enzymes 1, 2, 4, and 5 are mainly involved in the DNA repair part of the genome, and particularly DNA polymerase 1 is involved in Okazaki fragment processing, nucleotide excision repair, and some aspects of replication. However, the main polymerase enzyme which is responsible for rapid and accurate replication of genome in E. coli is DNA polymerase 3, which is our option number 3 in this case. Question number 51. The histones are tightly bound to DNA because, so the important concept in this case is that DNA is a negatively charged molecule. Therefore, it is supposed to be attracted towards a positively charged molecule. And in this case, in histones, it wraps itself around histones because of the positive charge present on the histones, which is our option number 1. Question number 52. Given below are two statements. Statement 1, a single gene can affect multiple phenotypic expressions. And statement 2, in a polygenic trait, multiple genes contribute to a phenotypic expression. Coming to statement 1, a single gene can affect multiple phenotypic expressions. This effect is known as pleiotropy, in which case a single gene can influence multiple phenotypic expressions and can affect a gene can affect two or more distinct phenotypic traits. So a classical example of this is the phenylketonuria gene, which affects multiple systems including our brain development, pigmentation and skin conditions. Hence this is a true statement. Coming to statement 2, it is in a polygenic trait multiple genes contribute to a phenotypic expression. This is true, and one of the best examples of this is human height, in which case multiple traits contribute towards the overall height of a human being. Therefore, both statement 1 and 2 are correct, and this gives us the option 1, which is the correct answer in this case. Interferon secreted by virus. Question number 53. Interferon secreted by virus infected cells induce. First of all, interferons are secreted in response to viral entry into a cell. Interferons are They are a form of adaptive immunity and help the body fight against viral entry or any other such disturbance. So so interferons when secreted by virus infected cells are supposed to induce an antiviral response, which will ultimately help our body fight against the entering pathogen. Therefore, the correct answer is the option 1, which states that it is an antiviral response. Question number 54. Lung cancer can be detected at an early stage by 1. Bone marrow test, 2. Blood test, 3. MRI scans, 4. Gene sequencing. First of all, option 1, bone marrow test. It is not recommended for lung cancer since it rarely changes the stage of cancer in non-invasively assessed patients and has no impact on tolerance of chemotherapy and barely affects the survival of the patient. Secondly, blood test. While blood test tells us about the overall health of the patient, it is not very specific for lung cancer and therefore again not recommended for lung cancer detection. Further, coming to MRI scans, this is one of the best techniques that can be used to provide morphological and functional data with diffusion, weighted, and perfusion sequences. It is currently recommended in the assessment of lung cancer extension to the chest wall and the mediastinum as a pre-surgical evaluation. So this seems to be the most ideal option for lung cancer. Furthermore, while gene sequencing provides a very promising avenue for testing lung cancer, however, biopsy samples are rarely enough to go ahead with gene sequencing. Therefore, the best option for lung cancer detection of lung cancer at an early stage is provided by MRI scans, which is the third option. Question number 55. Which one of the following is an autosomal dominant disorder? 1. Sickle cell anemia, 2. Cystic fibrosis, 3. Tay-Sachs disease, 4. Huntington's disease. Sickle cell anemia, cystic fibrosis, and Tay-Sachs disease, all three of them are known to be autosomal recessive disorders in which both copies of a defective gene are required to let the phenotype of the specific disease take place. And the fourth option, Huntington's disease, is an autosomal dominant disorder in which there is a even if there is a mutation in one HTT gene uh, out of two, it will lead to Huntington disease, which is a neurodegenerative disorder. Question number 56. Which of one of the following statements correctly describes type 2p restriction enzymes? A. They cut nucleotides only from the ends of the DNA. B. They act on only palindromic nucleotide sequences in the DNA. C. They recognize only GC rich sequences. Or D. they recognize only 80 rich sequences. We have to tell the correct answer from the options given below and which of the options are correct in case of type 2p restriction enzymes. So first of all, type 2p restriction enzymes are a class of restriction endonucleases that cleave DNA with the recognition sequences, generating either sticky or blunt ends, and they are known to recognize specific 4 to 8 base pair palindromic sequences within the DNA. So statement A, which states that they cut nucleotides only from the end of the DNA, is wrong, since the cleavage can happen immediately or within adjacent to the recognition sequence. Statement B is correct since it acts only on palindromic nucleotide sequence of 4 to 8 base pairs. And statement C and D are again false because they do not recognize GC rich sequence or AT rich sequences specifically. Therefore, only the B option is correct, giving us our correct answer, which is option number 2. Question number 57. Match items in list 1 with items in list 2. As you can see in this table, the list 1 contains incomplete dominance, recessive epistasis, dominant epistasis, and codominance. So we have to list the items, match the items of list 1 with list 2. In case of option A in incomplete dominance, the very famous example given to us is of flower color in 4 o'clock plant, which shows 3 different phenotypes depending on the genotype combination. Second, we have recessive epistasis, and first of all, epistasis is the genetic phenomenon where one gene's effect is modified by another gene. In recessive epistasis, we see that a homozygous recessive condition in one locus can mask the effect of gene on another locus. And one of the most famous examples of this is the coat color in mice, in which case the one of the most famous cases is the coat color in mice, in which the recessive gene for albinism, when present in homozygous condition, masks the effect of the of the gene on other locus, which gives the pigment color, therefore leading to albinism. So, recessive epistasis. Similarly, in dominant epistasis, the heterozygous condition or presence of a dominant allele even on one locus can mask the effect of the gene which is on the alleles which are present on the another locus. And fruit color in summer squash is a common example of this. The fourth option, the fourth uh, condition is the codominance, in which the two different alleles show effect at the same time, as is commonly known in case of ABO blood types, where if IA and IB alleles are present, resulting phenotype AB, and both the alleles are showing their phenotype in this case, which is known as codominance. Therefore, the correct option is a combination of list A and list 2 items would be A2, B3, C4, and D1, which is a fourth option. Question number 58. Given below are two statements. Statement 1. Activation energy is the difference between energy level of substrate and its transition state during this reaction. Statement number 2. Enzymes increase the activation energy, enhancing the rate of reaction. As you can see, this is a graph between the energy and the process of reaction. So when there are reactants which end up giving a product. They start on the level here and here. This energy is known as activation energy. This is the minimum energy required for two chemical reactants to react with each other, and it is also the difference between the substrate and its transition state, which is here. This gap is known as activation energy.